минут на 10 раньше закончу, вот у меня группа прям впритык к лекции.
Хорошо.
Поэтому я чуть-чуть раньше закончу.
Хорошо.
So, people, guys, start coming, start connecting with the lecture hall to hear a lecture. And today's immunology lecture. And today we discuss few issues. Aderinola, good afternoon. You are the first one today. And Sexina is the second one, so the others are coming. Okay, guys. Very good that you are coming a bit earlier. Okay, so today's lecture is in immunology. And today we will discuss some issues on the antigens. What kind of antigens are found in the viral and bacterial cells? But we are not going to discuss this topic in very details. Because as far as I know, we have discussed it in the classes already. So we go ahead with the topic, processing and presentation of the antigen, because that's not absolutely clear. For example, in my group, I didn't manage to discuss it properly. And then we go on for humoral immune response development. We take a look at the cells that are responsible for antibody-mediated immunity, and we try to discuss at least basically how it occurs. Understood? So now we go ahead with the antigen, and you know that this is a very important term that we use in immunology. And I would like to underline that. So antigen is not the same with pathogen-associated molecular patterns. Antigen is the unique, very small fragment of the molecule that will react with the antibodies or with T cells. So while the pathogen-associated molecular patterns are large pieces of the bacteria, viruses that common in different types of bacteria, viruses, and can be recognized by TLR receptors. So today, when we are talking about antigens and the mechanisms of their recognition, we discuss the much more specific level of self, non-self recognition. I'd like to remind you that in innate immunity mechanisms, we have discussed this as some level of recognition, but this level is of very low specificity. Now we see the very high level of specificity. That is the interaction between antigen and antigen recognizing receptors. And the only T and B cells may have antigen recognizing receptors only. Other types of cells, they don't have these types of the receptors. So that's why when we are talking and when we will be discussing the cells that participate in acquired active immune response development, humoral or cell-mediated, we can call them immunocompetent cells. What does it mean, immunocompetent cells? These are the cells that have receptors to recognize the antigen. And these I just want to repeat, just T cells and B cells, nothing else. So come back to the immunogen. And we have discussed already that it's supposed to be foreign to the human body. It is a foreign, so foreignness is one of the most important property of it. Then we discuss the property antigenicity or specificity, because as soon as antibodies are produced, they will react with antigen specifically. And the third very important property is immunogenicity. And regarding the presence of those properties, we divide all antigens into complete antigens and incomplete antigens that are called haptens. So we discussed that incomplete or antigens or haptens are not immunogenic themselves. It means they are foreign, they are specific, so they have antigenicity, but they don't demonstrate immunogenicity. Why? So haptens are non-protein molecules. Because they are not proteins, they cannot be processed by antigen-presenting cells and cannot activate T helper cells. So then, because they are small univalent antigens, they cannot activate B cells by themselves. So that's why they cannot stimulate immune response. So here you can see the examples of haptens. For example, beta-lactam drugs, penicillins, and cephalosporins are haptens. So they act as haptens. So they may react with antibodies if they are developed, but they cannot alone cause the development of the antibodies. So they need to be fixed, for example, with the complete antigen, that is the antigen of different biochemical structure. So now let's take a look at the possibilities. Let's take a look at the antigens from the point of view of antigen presenting cells. How effectively they can activate antigen presenting cells or some other cells. So it appears that the antigen that demonstrate a greater number and variety of different specific sequences that will exactly react with antibodies, and those portions of the antigens are called epitopes. For example, here you can see two types of epitopes are found on the surface of this antigen. A epitope here and B epitope here. So this is really not so diverse antigen. So here we can see the multivalent antigen with repeated one-type subunits or epitopes. This contains only one type of the epitope. So the same specificity antibodies will be binding with it. So, and the same specificity of antibodies will be produced against this antigen. And two types of antibodies will be produced against this antigen. So, and this antigen, this is a polyvalent antigen. So, with a variety of the epitopes, most probably this is a protein molecule. Can you see how many epitopes can be found on the surface of this antigen? So, and then against each epitope, a special antibody, specificity antibody will be produced. So a variety of antibodies with different specificities can be produced against this single antigen. So these are more immunogenic antigens. So then if we take a look at small univalent, generally the least immunogenic, that is the example of HAP10. So this is the least immunogenic because it is not large. It contains not, it doesn't contain not repeated subunits. It has the only one that is really very demonstrate very poor immunogenicity. That's how HAP10 appears. So then we mentioned that HAP10, when they bind with the carrier protein, then immune response can be produced against this antigen. So now let's take a look at the antigenic determinants or epitopes. What are they? So these are smallest units of antigenicity. So this is the glycoprotein. So if we put it all together, this is a glycoprotein antigen. But it consists of the polypeptide chain with very special conformation. And we can see a lot of epitopes on the surface of this protein molecule, and they are all different. So the smallest chemical groups that will react with antibodies and against which the antibodies will be produced are called epitopes. And usually let's come back to this picture. So some may have very few epitopes, 2 or 1, identical, and some may have many different epitopes with a different specificity. So, and if we take a look at the virus, and this is the envelope virus, the most important epitopes usually are on the surface of the glycoprotein somewhere that are responsible for binding with the cell surface receptor. If we take a look at the bacterial antigenic determinants, so you can see that bacteria usually carry much more epitopes. So because they are much more diverse in their structures. And the epitopes or epitope is capable of sensitizing an immunocyte and reacting with the complementary site in a specific antibody. So, for example, this is the antibody against this antigen, but then so you can see that out of all antigen, they will react with the particular epitope, right? So, and the microbes may have really different epitopes, and the specificity of the antibodies produced against the microbes will be different, and the specificity of the antibodies uh, depends on the structure of the site in the immunoglobulin molecule or antibody that matches with structurally with the antigen, with the with the epitope of the antigen. And the structure in the antibody is called paratope. So epitope is on the surface of the antigen and paratope is on the surface of the antibody, and they react together specifically. And usually the antibodies that are directed against particular epitope will not react with a different epitope. So this demonstrates and you can see if we take a look at the antibodies. So they consist, or monomeric antibody consist of long chains and short chains. 2 long chains that are called heavy chains. See, this is the heavy chain and this is the heavy chain. So it has two heavy chains and two light chains. And in both heavy chain and light chains, we have so-called V region, variable regions. Those regions are responsible for binding with a particular epitope of or antigenic determinant. So we have the interaction against the space that is here and the epitope of the antigen. So if we take a look at the viral antigens, so you can see that any parts of the viruses may be antigenic or immunogenic. The capsid proteins and the nucleic acids are also can be also antigenic. So, and if we take the envelope again, nucleic acids, the capsid proteins, and the envelope proteins are antigenic. But of course, the protective antigens are those that will be responsible for binding with the cell receptor, because antibodies against this exactly epitopes, so will prevent the infection. Now the question is, if the antibodies are produced against viruses, then how these viruses will be damaged? That is really a question. What will be the function of these antibodies? Because, so, this is virus-specific antibody. Take a look. IgG molecule, let's say. So, it may kill nobody. It is not developed for killing. So then the only thing the antibody can do is to bind with the virus antigen or bacterial antigen or whatever antigen, right? If we are talking about the viruses, so it binds with the antigen like this only. That's the only way what they can do. Just bind with the antigen or with its particular epitope. But then when this antibody binds with a particular epitope on the viral antigen, this complex may activate several lytic mechanisms, and these lytic mechanisms are shown here. For example, this antigen antibody complex may activate the complement system. What will happen if the complement system is activated? Membrane attack complex is formed, and uh, mm, the molecules of membrane attack complex, the proteins, will be embedded into the membrane, so they cause destruction of this cell, of the virus with the help of antibodies. Then the second way, virus-specific antibody binds with the virus, right? So then the Fc portion is free to bind with so-called Fc gamma receptor shown or display or demonstrated on the surface of macrophages and neutrophils. So what does it mean? So this system will be activated and this lytic system will be activated. So this is the second system, or these are the proteins that will be activated with the help of antibodies to kill the virus or virus infected cell. So then, except for this, we have one more way that is called the NK cell activation. And altogether, these processes are called antibody-dependent cytotoxicity. So, ability of lytic mechanisms in our body to be activated with the antibodies. Now let's take a look at bacterial antigens. And again, we have discussed them during the classes. So, but these are mainly gram-negative bacteria antigens, right? So then we also have a unique antigen that can be found on gram positive bacteria, but not on the gram-negative bacteria. Like, for example, teichoic acids, right? So, or some lipids incorporated into the cell wall of mycobacteria, for example, right? Okay. So, but then the question is how the bacteria can be eliminated. My dears, are they very much safe? So, we have limited number of lytic mechanisms that depend quite often on the presence of the specific antibodies. And if bacteria are opsonized, this term is used to explain that bacterium-specific antibodies cover or surround the bacteria, right? So then these specific antibacterial antibodies may activate the complement system to produce membrane attack complex and to uh, react with the Fc portion of the immunoglobulin or of some C3b molecules, and the macrophage usually react with the Fc portion of the antibody, and neutrophils are usually react with the C3b molecule that comes from the complement activation. Do you remember this? We have discussed it. So, and then we have the effective killing of bacteria. So now let's take a look at the development of the immune response in a body. But we will not talking about adaptive immunity, we have discussed it. We will be talking, innate immunity. We will be talking about adaptive immunity. And then again, we have B cells and T cells here. These are the only immunocompetent cells. But usually, at least T cells, they need help from the innate immunity mechanisms, from the dendritic cells mainly and phagocytes. And you can see how phagocytes activate so-called T-naive cells. What cells, what lymphocytes are called naive or naive? Those that never meet their antigen yet. So it means they are not fully differentiated. So they undergo non-specific differentiation uh, that doesn't depend on the antigen. But then they will be activated when they meet their own antigen here and here through the macrophages. And here you can see that B cells may recognize antigen directly by its receptor, E antigen recognizing receptor, and T cells cannot do this with the antigen recognizing receptor. The antigen should be presented by antigen presenting cells. Then these cells proliferate, B cells and T cells, and then B cells start producing, differentiate and produce antibodies that eliminate the microbe. And T cells will differentiate into several groups, and one of the group will be called effector T cytotoxic lymphocyte that also may eliminate the microbe from the body. This is a basic scheme. So now we take a look at the B and T cells, but before we do this, we have to know and exactly underline what molecules on the surface of the lymphocytes and some other cells participate in this interaction. So we're supposed to talk about clusters of differentiation or CD markers. So what are CD markers? These are surface antigens or surface markers, surface protein molecules of human lymphocytes. They are identified with the help of the usually immunofluorescent test or ELISA test with the help of so-called monoclonal antibodies. So how it happens? It means that every surface protein molecule that has names here, right? So can be taken out of the B cell and injected into the body of the animal. Then B cells will be taken from this animal that already get information about the particular antigen. So then the antibody should be produced against one out of one single B lymphocyte. I hope you studied the uh, method or technique of producing monoclonal antibodies in biochemistry. If not, when we discuss antibodies, so in the classes, we'll discuss also monoclonal antibodies. But the thing is that these antibodies will react highly in a very specific way with the only one epitope on the surface of particular CD surface marker. So, and if we couple these antibodies or conjugate these antibodies with the immunofluorescent dye or enzyme or whatever, we make the interaction between the antibody and the antigen visible. So that's how we can find the particular CD markers on the surface of any cells, including B cells and T cells. And now you can see that B cells and T cells are very similar. And uh, quite often they say that microscopically they are indistinguishable because they have really very common morphology. But they can be recognized by the markers or CD molecules that are found on the surface of these cells. So let's take a look at the B cell. And today we mainly be talking about B cells and not so much about T cells. When we discuss cell-mediated immune response, then we will be talking mainly about T cells. So what is the most important receptor on the surface of the B cell? You can see it here. It is called B cell receptor, and it is shown here as immunoglobulin molecule, and it is immunoglobulin molecule, and this receptor function is antigen recognition. And on the surface of the B cell we may have many of these immunoglobulin molecules, but their structure is absolutely identical. It means the CD molecules or the immunoglobulin surface molecules that are called B cell receptors on the surface have the same specificity. So they are of single antigen specificity. Then other important molecules are, for example, molecules CD35 and CD21 that are receptors for different components of the complement system. So we mentioned that some components of the uh, complement system may activate B cells through binding with these particular receptors. So then, apart from this, we may demonstrate the receptor that is called CD40, CD marker. And the CD40 is the signal or surface receptor that binds the soluble signals produced sometimes by some of the T cells, Th2 cells, so-called, that generate humoral immune response, And those signals, if they bind those protein molecules with CD40 molecule on the surface of the B cells, that may cause activation of B cells to proliferate and differentiate. So they are it is a stimulatory factor, or it is responsible for stimulation of the B cells. And finally, we take a look at the antigen that is found on any antigen present in cells. Now we'll talk about them. We'll talk about them a bit later. Uh, so this is the molecule that is associated with antigen present in cells only. So, and of course, the function of MHC2 is the antigen presentation to T cells for their activation. And we won't discuss so much the T cells, but we just mentioned. Of course, they also have some very important markers. We just mentioned one very complex receptor. So this receptor is made up of T cell receptor, and of course this is an antigen recognizing receptor. But this receptor is tightly fixed with the so-called CD3 molecule. So, and it makes a protein complex with the TCR receptor. Why? Because this is the CD molecule that help the transducing of the signal produced from antigen recognition on the surface, and the activation should be transduced deep inside the T cell nucleus. So they only then the cells start to be activated. So CD3 is responsible for signal transducing from the membrane into the nucleus. And TCR is responsible for recognizing this signal. So, and these two types, T cells and B cells, will produce different types of immune responses. So, humoral antibody-mediated immune response and cell-mediated immune response. Why do we call them like this? Because in a humoral immune response, as a result of some T and B cells activation, the B cells will differentiate into plasma cells and secrete antibodies. That is the result. They will protect us against the different, usually extracellular pathogens, because they bind with them and opsonize them and activate the lytic mechanisms in the body. So, because these products are soluble in the serum, so we call it humoral immune response. And the second immune response that depend mainly on different types of the T cells. So it is responsible for cell-mediated immune response that is much more effective against so-called intracellular parasites. I'd like to remind you, what does it mean, intracellular parasites? So these parasites get inside the cell, so they are protected from antibodies by intracellular location. So to get rid of this pathogen, the whole cell is supposed to be killed. So that is the idea of this type of immune response. So that the cell infected with the intracellular pathogen is supposed to be killed. Or also cancer cell that is recognized as a foreign cell supposed to be killed within this type of immune response. So now we take a look at the antibody synthesis and humoral immune response. uh, if we take a just rough look at this, so the antibody synthesis need three types of cells to be involved in the process of immune response development. And first group of cells is called antigen presenting cells, or APCs. And APCs are major APCs, macrophages, lymphocytes, and dendritic cells. Then the second type of cells that need to participate in this, these are one of the subpopulation of T helper cells. So we call them T helper cells because of their function. They are not really uh, effect have high effect, so they are not effector cells themselves according to their function. According to their function, we call them regulatory cells. So those T cells that participate in this immune response are called regulatory T cells. But not all T cells are identical. So the subpopulation of T cells that participate in antibody response are called T helper 2 cells or Th2. How they really work? So Th2 cells produce very important, very strong cytokine signals. So many cytokines can be produced by these cells, but the major one is interleukin 4. And these cytokines activate B cells for proliferation and differentiation. And as a final result of differentiation, the antibodies will be produced out of the uh, B cells, so they are plasma cells. And the antibodies start acting. So, and then here you should understand that cells, when they develop immune response, need to cooperate together. So they need to do something together, to cooperate together. So, and then you have to understand that these cells cooperate together in two distinct ways. So the first way of cooperation is shown here. This is the signal. So the antigen presenting cell should touch the T helper cell through the receptor and the common foreign antigen that is presented by APC and recognized by T helper cell. So this cooperation is touching. So cells need to come in contact directly. So this is called cell-to-cell contact. And the second way of their interaction is through cytokine release. So it means, for example, here, macrophage, when it is activated by process of antigen processing and presenting, start producing signals, interleukin 1, interleukin 12, and tumor necrosis factor. These are mediators that may activate T helper cell to come in contact and to recognize the antigen. So this is mediator cooperation. And if we take a look at uh, the process of this is something like smelling of some molecules in the air, right? So this is touching, and this is smelling of the molecules in the environment. So this is cell to cell and mediator or cytokine cooperation between different types of cells participating in the development of the immune response. And we go on again. We would like to take a closer look at B cells. So let's take a look. Okay, B cells. So B cells may have many different types of important receptors. So, for example, let's take, and you're supposed to know some of those receptors because by them we may recognize them. So, I would like to pay your attention at the receptors that are called CD90, CD20, CD21, and 22. These are the cell receptors, or CD receptors, by which the B cell is recognized. So CD19 receptors is expressed in all B cell lineage, right? Except for the plasma cells. It means that within the whole way of differentiation of B cells, this receptor will be visible. But then, if the B cell finally differentiate into plasma cells, then plasma cells disappear from the surface. So this CD19 molecule works as an adapter protein to recruit cytoplasmic signaling proteins to the membrane and to work within the B cell nucleus. So it is for signaling about the binding with the surface receptor. Then the complex CD19, CD21, the decrease the threshold for B cells receptors signaling pathways. Then CD20 is a membrane, and you can see it here. CD20 is a membrane embedded surface molecule that is very important in the differentiation of B cells into plasma cells. So on the final stage of differentiation. So, okay. So what else should we know about the B cells activation? So if you can see here B cell receptors. When the B cell receptors bind with the bacterial cell in the diagram or any other pathogens, the B cell receptor becomes cross-linked. It means one large uh, particle with identical antigens, for example, lipopolysaccharides, right? So cross-link the neighboring receptor, and by this the very strong signal will be produced. And this very strong signal will be the first step in B cell activation, because this integral or the intracellular tails of the proteins will be responsible for signaling into the nucleus. This may occur in the regional lymph nodes near the site of infection. So the pathogen gets to the lymph nodes via lymph, and then when B cells are activated, so that will be the first step. And here I would like, because that depends on the B cell activation, I would like to draw your attention that in humoral immune response development, we may distinguish two types of antibody immune response. TI, T independent immune response, and T dependent immune response. I would like to repeat that both are humoral immune responses. But then these types of humoral immune responses depend on how B cells are activated and what kind of antigen participate in this activation. So first you see that this is the first one is T cell independent pathway. So, and this pathway is very typical for T-independent antigen, for example, lipopolysaccharide or any carbohydrate antigens that don't contain proteins. So those carbohydrate antigens usually univalent or very univalent, right? So we take a look at this. I would like you to associate this with the antigens. Oh, here we are. Can you see? That's what kind of antigens are we talking about? This or more probably this one. That has one specificity epitope that is repeated many times. Okay, get it? Oh, see, those identical molecules, the identical epitopes, bind with BCR receptors located on the distributed on the surface of the B cell because they are same specificity epitopes, so they bind with many cell surface B cell receptors, and the activation is so strong that B cell, without any other signals, just because of antigen recognition, can be activated to produce plasma cell that produce immunoglobulin M. But my dears, this is the only result of T-independent immune response. So the only immunoglobulin M will be produced, and the differentiation of the B cells into memory cells will not happen. Why it is so? Because here you can see no signals from any good regulatory cells, T regulatory cells are produced. And for full humoral immune response, those signals from T cells are needed. So if those signals are obtained, B cell may get this signal. Then the differentiation may happen in the plasma cells and memory cells. And then plasma cells may be producing many classes of antibodies, not only IgM. What's wrong with IgM? They are very good antibodies, but they are short-lasting antibodies. They will wane from the blood very quickly, so they may give limited short-term protection for a body. And now let's take a look, because here is the something that is in between. And here let's take a look at the B cell that may act together with the uh, protein molecule. So this is the carbohydrates with protein molecule. While B cell is activated, because B cell is antigen presenting cell, it can internalize all this, the whole molecule. Then it can process this molecule and present it on the surface of the cell together with MHC2. So, and also it may produce some and the T cell will be activated by recognition of this presented epitope of the complex antigen. So T helper cell will produce cytokines that will be activated additionally B cell. So the B cell will differentiate into plasma cell plus into memory cells. Apart from the memory cells that will keep memory about their offended antigen for a very long while, sometimes long for the rest of the life. But then the uh, plasma cells can produce not only IgG, but they also can produce IgG, IgA secretory antibodies and the others. So the immune response will be much more strong and it will last longer. Apart from this, the memory cells will be produced. Okay? So, and here we may see when the only peptide antigen is digested, the same H cells will be activated to produce the full immune response. Okay? So then now we can see that this is the function of the B cells during humoral immune response development. So the B cell, these are the B cell receptors, and this importance is antigen recognition. So once they bind directly with bacteria or viruses, recognizing particular epitopes with their unique specificity T cell receptors. This, you see, this is a sign of activation. So antigen binding causes the activation of B cell. My dears, everything in the body, if we are talking about immune response development, starts with antigen recognition. So this is a very first step. So why it is so important? There are many B cells in the body, right? And we call them resting B cells. So they all have unique BCR receptors. So when bacteria with its antigens enter the body, it's no need to activate all B cells because it will be chaos in the body. Then only those B cells will be activated that recognize the antigen that right now present in the body. And the rest of the B cells will not be activated. And this is very wise because they don't need to be activated because they are directed with their antigen recognizing receptors, not against bacteria that enter the body. So once this particular clone of B cells will be activated, then what happens? So MHC class 2 will be fully expressed on the surface of this B cell. And also it starts producing something new. What is new? So this is receptors for cytokine C. Before the activation, they did not have these receptors. So now they have. That is the point that make these B cells susceptible for further activation. Those cells that don't have contact with the antigen will be not able to be activated. So then, as soon as this cell is activated, it produces the peptide and activate B cells. And here we have CD40 molecule important for additional activation. So, and these activated T helper cells start signaling through this interaction with these co-stimulatory signals. So, and then again, the cells start to be activated and bind with the cytokines, and it increase in size. And then, as a result of this, we will have B cell clonal expansion and differentiation. How it happens, really, I just want you to take a look at the scheme, how it happens in the at least scheme, how it happens in a body. You can see here steps of antibody production. So we can see B lymphocytes with the antigens bound to the B lymphocyte through their surface receptors. And the surface receptors are usually immunoglobulin molecules of classes of classes M or D. It means the structure of the surface molecules is identical to monomeric IgM molecules, and they are fixed tightly with the cell membrane with their Fc portions. Then B lymphocytes may perform so-called processing and presentation of the antigen. And they perform the processing and presentation and may present then the antigen to T helper cells. And T helper cells here, T helper 2 in our case, they may produce interleukin receptors, and major are interleukin 4 and interleukin 5. And this is demonstrating two types of cooperation between these two cells. The cell-to-cell cooperation directly, right? And mediator or cytokine mediator cooperation, so touching and smelling of some signals. Then B cells start to be fully activated, proliferate and differentiate into plasma cells and memory cells. So memory cells are long-living cells. They will stay in a body for a very long while. But then they keep on the surface these BCR receptors identical to what the initial resting B cell had, right? So they all are of the same identity, these antigen binding receptors. So that's how the memory cell keeps memory about offended antigen. Its surface receptors will bind with only this antigen but not any other antigens. And plasma cells differentiated from the same clone lymphocyte. They first start producing the immunoglobulins that already once produced by this cell. So it means the IgM molecule. So usually IgM antibodies are produced first. I think, do we have? Okay. And then except for IgM, they produce IgG, IgA, and so on. So by switching under the signals of T helper 2 cells. So then class switching, that is the ability of the plasma cell to produce first IgM and then IgG and IgA or some other immunoglobulins. Now, again, I would like to remind you, what does it mean, clonal selection? You're supposed to hear, so maybe you heard about it when you have studied, uh, mm, I think, cytology and histology, because this is the quality over the B cell clones and T cell clones. While we were talking about B cells, you can see here. So this is a stem cell, right? And they differentiate into pre-B cells. And then they mature and differentiate into B cells with B cell surface receptors. So each cell is a clone, right? So because each cell has its own specificity immunoglobulins, so the clones. But then they appear in a body in not so activated state, right? Why they are not activated? Because they don't get the signal about antigen recognition yet on their surfaces. See? They all equally rest in cells. But then bacteria virus enters the body. And antigens are released. So, and this antigen fits with this immunoglobulin receptor of this B cell clone. So the fact is that other cells will not be able to be activated. So the only this cell clone or few B cell clones will be activated. So now it is activated and stimulated, and this stimulated cell start proliferation and differentiation into plasma cells and dendritic cells, and memory cells. So as a result, we have few B memory cells and many plasma cells. So this ability of one or several clones to proliferate and differentiate quickly regarding the meeting with the antigen is called clonal expansion. It means out of many clones, the only one B cell clone get activated and proliferate and differentiate, and the fact that it is done in the body is called clonal expansion. So the B cells will be activated, able to recognize the particular clone of the antigen. And of course, they produce antibodies against this particular antigen only. And then when the antibodies will be produced, they bind with the bacterial cell to mark them for other lytic mechanisms, and the eliminating of the microbe will be produced. Okay? Okay, then now, humoral immune response can be divided into primary immune response and secondary immune response. What does it mean, primary immune response? Let's take a look at the B cell and the activation of several B cell clones and their proliferation and differentiation. At the very beginning, they start producing immunoglobulins M, right? And then later on, so when we perform class switching and the T helper cells will be activated, they start producing memory cells and also IgG antibodies. That's all together is the primary immune response. So, and because the body is not familiar with the antigen, that is what we call primary immune response. When the antigen enters into the body, that never happened to be in this body, so unfamiliar antigen. So, but then if it happens, then the immune response has really lag phase or lag period. Why? Because this is a time needed for antigen binding, processing, and presentation. So for B cells activation, for clone proliferation and differentiation. So the lag phase or lag period occurs within 5 days. Only after 5 days, some antibodies here, right? First IgM and then IgG start to be accumulated in a body. And then take a look here. First, IgM will be produced, right? But then they appear but very quickly disappear from the blood. And while they start disappearing, the IgG antibodies start to be produced. Usually, the concentration will be higher than IgM, and then they also stay longer in the body, but then within time they also disappear. So that is the primary immune response. It's over. What is the result of immune response? For example, after 1 or 2 years here, right? Immunoglobulins may not present in the body anymore, but memory cells are present. Take a look here. They survived and live for a very long while. If, for example, after 2 or 3 years, the same antigen enter the body, then the memory cells will be activated directly. So we will not have any delay in antibody production. The first antibodies will be produced within 1-3 days. So we have, because the way now is very short, right? Here the way was very long. Here the way is very short. So first, the immunoglobulins M will be produced by, please pay attention. The same amount of immunoglobulins M will be produced. But then the IgG immune response will be much higher and long lasting. And by the way, more memory cells will be produced, and more memory cells will retain after down regulation of the immune response. So it means that even when we are talking about adaptive humoral immune response, this immune response may be improved by repeated injection of the antigen. Can you imagine if this is a vaccine? So first, when we inject vaccine, it takes longer time to produce antibodies and memory cells. When antibodies disappear, a few memory cells survive. And for example, we perform vaccination again second time. So more antibodies are produced, and of course more memory cells will be generated in the body to survive for a very long while, to give protection even after many years if the same antigen as a normal infection, natural infection, will appear in the body. Now let's take a look at the process of antigen presentation and processing. Let's take a look first at the antigen presenting cells. So the altogether 3 types of cells are considered so-called professional antigen presenting cells. These are dendritic cells and macrophages. These are similar types. And the different one, that is a B cell. What are the most important features of them as professional antigen presenting cells? So they are capable of phagocytosis, and this is usually dendritic cells and macrophages, or even rather macrophages, and internalizing of the antigen through receptor-mediated endocytosis through the recognition by B cell receptor. So it means these cells, professional antigen presenting cells, can so-called internalize antigen by different ways. One way is phagocytosis, and the second way is endocytosis, okay? So then, some of the antigen presenting cells may express receptors for apoptotic cells, damage-associated molecular patterns, and pumps that are molecules on the pathogenic microbes. So, do you remember how do we call those receptors? T cell receptors here. So, but B cell doesn't have toll-like receptor. It doesn't have toll-like receptor. B cell receptor is enough for binding with any viruses or bacteria. Then what is underlined, please? because this is really associated with the function of antigen presenting. So the cells that you see here constitutively express high level of MHC class II molecules. And this one also. Constitutively, it means constantly. It means the expression of MHC2 doesn't really depend so much on the environmental signals, interleukins and cytokines. They keep them always on the surface. Why it is so important? Because MHC class II molecules are those molecules that's supposed to be bound with a foreign antigen and express to present on the surface to present the antigen to T cells. And also what is important, uh, all these types of cells express co-stimulatory molecules when they are activated by the antigen recognition or pumps recognition. That's also a very important property. Okay. That's interestingly, we may have atypical or non-professional antigen-presenting cells. And you may see here, mast cells, basophils, eosinophils. Okay, that's too specific. We won't discuss it. So what they don't have MHC class II molecule normally, but they may express this molecule under some signals. Inducible, it means if they get some interleukins, they may start producing MHC2, so they may act as additional antigen presenting cells. Okay, now you've seen this picture in the protocol, right? So, and I just want to remind you that this is one of the most important antigen presenting cell. This is a macrophage. So, and I would like again to tell you that macrophage may bind with the microbe through many different molecules. For example, through TLR receptor, then through CD14 receptor. Then they may bind with a microbe with mannose receptor. Because do you remember that mannose is the carbohydrate residues on the surface of the bacterial cell or fungal cell wall or bacterial cell wall? Then macrophage may use its complement receptors to bind with the opsonizing CD3B, oh, sorry, opsonizing here, sorry, CD35 and CD11, so opsonizing molecules. And also, macrophage have receptors that may bind with the Fc portion of the immunoglobulin G if it is bound with the antigen. So it means the macrophage can be activated by many ways. So do the dendritic cell. The dendritic cell also may have T cell, toll-like receptors right here for the partons of the pathogens. They also may bind the C-type, so through the receptor to C-type lectin that also cause the signaling and activation of this receptor because it binds with the carbohydrate structures on the pathogen. So then, while activated by the antigens, the signaling should occur through NF-kappa factor for co-stimulation of adhesive molecules and cytokine production. And the dendritic cells start to be able to express MHC class II on the surface. Together, you can see here with the foreign antigen as it is processed and expressed on the surface. Then who they present the antigen? To the T cell with its most important antigen recognizing T cell receptor. So this is a function of T cell receptor to recognize foreign antigen. And moreover, the dendritic cells are important in cell-mediated adaptive, immune response, because depending on through what kind of the receptors the antigen presenting cell is activated, that's define the spectra of the signals, interleukins or cytokines produced by antigen presenting cell. And it appears that antigen presenting cells may produce signals to activate T helper 1 differentiation from the T helper cell, or they may produce the signals that activate Th2 type of T helper cells proliferation. And you can see here that if the T cell that is not that is called T naive cell and never had any contact with the antigen, depending on the antigen and what is the signal produced by antigen presenting cell, may differentiate into Th1, and it produce the Th1 will produce the gamma interferon and interleukin 2 that would activate cell mediated immunity, or depending on the particular signals, Th2 cell will be activated, and it produces signal interleukin 4, 5, and 13 to activate B cells. So this will be cell-mediated immune response very high, and if into this, this is called polarizing signal. If polarizing signal goes into Th2, so then interleukin 4, 5, and 13 will be produced for humoral immune response generation. Then I would like to talk about the antigen presentation, because we haven't discussed it in a class. That is the very important thing to understand also. So why it is so? Because first way we discuss is the MHC class 2 pathway. How the cells in our body may may present some antigens on its surface. So first we are talking about antigen presenting cells, because the only antigen presenting cells may express MHC class 2 on the surface. So, and it means they may activate the T helper cells for their activity. But this is called exogenous pathway of activation. Why exogenous? Take a look here. So we have bacteria or virus or their particular or bacterial toxins or whatever, any protein molecule that is foreign for this antigen presenting cell. This antigen presenting cell engulf this antigen or engulf this bacteria or virus. Then phagosome is formed, and then phagosome is fused with the lysosome to produce the phagolysosome and for the digestion of the molecules. And out of those molecules, a unique peptide will be developed. Unique peptide. So this peptide usually made up of 15 to 30 amino acid sequences, not more. It is quite small. So then, what happens in the body? This is the rough endoplasmic reticulum, right? and you know this. And you know that the proteins are produced self molecules like MHC class 2. This is a self molecule, and it is expressed to demonstrate that I'm self cell. So this is produced in the uh, rough endoplasmic reticulum. But as soon as MHC class, this is a red one, produced, the antigen recognizing site is blocked with the so-called invariant peptide. Why this place in the MHC class 2 molecule is blocked? Because this is the place where the foreign molecule, foreign antigenic epitope should be uh, placed. So, and this is for prevention of our self peptides to be bound with this antigen binding site. So, this MHC class 2 molecule with invariant peptide now then will be uh, transported to the Golgi, and in the Golgi apparatus, in Golgi complex, all other fragments of these invariant peptides will be removed except for one that is called clip. So still clip, uh, other chains are digested, but the clip still will cover the antigen binding fragment or region in the MHC class 2 molecule because it needs to be replaced with the antigenic foreign peptide. So as soon as it is combined with the phagolysosome, so then the antigenic peptide developed from bacteria or virus will be immediately fixed with antigen binding site. And then this complex that is made already out of MHC class 2 and foreign antigenic peptide will be expressed on the surface of antigen presenting cell. So the peptide should be expressed together with MHC class 2 but not alone. The only then the T helper lymphocyte will be able to recognize this peptide. That is called exogenous pathway because the microbe enters from outside into the cell. But then we need to discuss what happens in the cell to be prepared for further, uh, to discuss some more events that occur in the human body and immune response development. We need to understand that MHC class 2 molecule is demonstrated on the surface of antigen presenting cells only. But then we have another MHC molecule that is called MHC1. And this MHC class 1 molecule is demonstrated or is presented on the surface of any nucleated cell in the human body. And MHC1 is so-called alloantigens. What does it mean? It exists in any human cell, but they are different in different individuals. So the structure of MHC class 1 is unique to each particular individual. So then what happens? And this is a not antigen presenting cell. So it means it can be any cell in a body, muscle cell, epithelial cell, endothelial cell of blood vessels, or it also can be the phagocyte, for example, because it will also produce MHC1 on its surface. Then now what happens if the cell is infected with the virus? You studied general microbiology. When virus enters, so it mm, release its nucleic acid And the nucleic acid, if it is DNA, it should be transcribed into RNA. RNA will be sent, mRNA will be sent to the ribosomes to produce viral specific proteins as endogenous protein. It means these proteins will not appear in the lysosome or phagosome or endosome. No, these proteins from the ribosomes will be released directly to the cytosol where nothing really happened with it. So then, what happens with the peptides that are produced on the ribosomes of the particular cell, of the cell? They are processed in the cell as self-peptides. So, endogenous antigen, so it means virus-specific proteins produced by the ribosomes in the cell, will be processed as if they were the self proteins. So they will be sent to the protosome and degraded into protosome like any other cell proteins will be degraded. And the peptide will be transported to the rough endoplasmic reticulum through the special transport TAP system, right? So where we have tapazine, beta-2 microglobulin, and calrectin and calnexin, right? And then here we will see the development of the unique peptide that will be transported to the MHC1 because MHC1 is usually here and it is not blocked with anything. Why? Because it is to bind with the self proteins. So the peptide processed through the protosome will be bound with the MHC1, but this is not self peptide. This is antigen specific peptide processed as if it was the self peptide. And then these will be transported to the surface of the cell and expressed on the surface of the cell. So again, we can see MHC class 1 peptide complex. Peptide is a foreign peptide molecule, viral protein here, right? Or viral epitope here. And this will be expressed on the surface of the cell. So this signal will be recognized by T helper cells, and this signal will be recognized by T cytotoxic cells. So these two ways of uh, uh, antigen internalization, processing, and presentation, will determine the way of MHC class 1 restricted recognition or MHC class 2 restricted recognition. So, and with these guys, I would like to finish. We were working without really any break, so then it will be enough for today. And next time we discuss the results of humoral immune response development. We take a look at the antibodies and their functions, and that is the topic partially familiar for you because you have studied it in biochemistry. And then we discuss what are the mechanisms over the microbes elimination from the human body with the help of antibodies. And then we go ahead with the humoral and cell-mediated immune response development. With this, I would like to finish. So, and next week will be microbiome week, so we have lecture in microbiology, and the week after the next week again immunology. Guys, stay home, wear your masks, treat everything around with the sanitizing, and if you feel not so good, please refer to the students polyclinic. Okay, if you need a doctor, go to the doctor. Okay, so good luck for you and good luck for those who are listening me from India. I know that quite a lot of you are sitting in the red zones. Please take care, stay healthy. Goodbye.